Awesome. Well, welcome everybody. Thanks to tuning in for another edition of Talking Trout. We got a pretty cool guest doing some really interesting research here tonight that we're excited to share with you. We have Kristina Pechacek. I think I got that right. Pechacek with us. She's a student at UW La Crosse, and she's been doing some really interesting research on how brown trout populations interact and compete with brook trout populations in streams in the driftless area. And it's something that was brought to the state council's attention, maybe a year and a half ago, I think, Kristina, and some of the folks at UW La Crosse were looking for some funding for this particular research, and it was something that I think the council put some dollars in, and I believe the local Coulee Region TU chapter also supported some of the funding for the research project. So we're super excited to have Kristina Pechacek here with us tonight, and I'm going to turn it over to you, Kristina, and take it away.
Okay, so I'm going to make sure to close everything out so you guys can see the full video of everything. So you can still see everything okay?
Yeah, it looks great.
All right.
Can I say one more thing before we get going?
Yes.
I think Kristina's got about a 35 or 40-minute presentation. And so what we're going to do is try to save all the questions for the end. So if you do have a question that comes up along the way, just pop it in the chat box, and I'll make a note of that. And we'll come back to those questions at the end of the presentation. So with that, the floor is yours, Kristina.
Sweet, thank you. Yeah, as I was introduced, my name is Kristina Pechacek. I'm a graduate student at the University of Wisconsin La Crosse, and I'm working on my master's right now, getting a degree in biology with an aquatic science concentration. And today I'm just talking about my research, which encompasses two different field seasons, the summers of 2019 and 2020. And my official title is Interspecific Competition between Brook and Brown Trout within the Wisconsin Driftless Region. Of course, I have to do an acknowledgement slide and thank everyone that have been involved in this project and made this project really possible. First, UWL for their RSCL grant funding, and UWL River Study Center for their financial and project support. Other graduate students and undergraduate students assisting in sample processing. And then my committee members for all their support and assistance throughout this project. Trout Unlimited for their funding. With several chapters. And then I want to especially thank the Department of Natural Resources, specifically Kirk Olson, Kevin Mall, and Troy Clement, because without their plans of the Brown Trout Removal Project and their efforts throughout these last few years, my research project really could not have been done. To give you just a general outline about everything that I'm going to take and talk about, I will be explaining a brief overview of brook and brown trout identification and distribution. Then I'll be moving into the probability of occurrence and climate change effects within the driftless region. Then I will explain the knowledge gap and the purpose of my research. And then I will get into details about my research project and some preliminary results that I've found so far. And then finally, I will just give a summary and some take-home messages from this presentation. Okay. To start with, if you don't know already, the driftless region is comprised of 4 states, Minnesota, Wisconsin, Iowa, and Illinois. And the reason why it was called the driftless region is because the area is absent of glacier drift. Glacier drift is comprised of deposits of silt, sand, gravel, and rock that is retreating glaciers left behind, hence the name the driftless region. They didn't drift over this area. Within the driftless region, there are 2 prominent salmonid species, the brook trout and the brown trout. And then these are the 2 species that I will be studying. So the illustrations here on the left are of the brook trout, and the illustrations here on the right are of the brown trout. And then some key points I want everyone to kind of take away from these species is that the brook trout is Wisconsin's only native stream trout species. And generally the brook trout is smaller than the brown trout, ranging from only 6 to 13 inches in length. And as you can see, the brook trout has some very beautiful, vibrant coloration patterns and a very distinct marbling pattern on its back called brumiculation. And then the brown trout was brought over from European descendants in the 1880s, and they are also been known to be called the German brown. And the European descendants thought to bring these brown trout over because it has similar climate within the US and for more food availability and angler opportunities. Secondly, brown trout are generally larger than the brook trout and can grow over to 20 inches in length within some of our inland streams. And as you can see, the brown trout can also have some very beautiful coloration patterns. And then finally, over the last several decades, brown trout have gained a large following and created a booming industry for trout anglers, especially fly fishermen, and created not only an ecological importance, but a large tourism industry. And then moving into more of distribution, to understand the distribution of brook and brown trout throughout the United States, maps were taken from the USGS website and it's current up to this year, 2021. What I want you to just kind of focus on is the orange and the maroon colors within both maps. The maroon indicates the non-native population areas and the orange illustrates the native population areas. When looking to the map on the left, you can see that the brook trout have non-native distribution primarily through the western side of the United States. And then you can see that they have native populations covering almost the entirety of the state of Wisconsin. And then when you're looking on the map to the right, all you see is the color maroon, which makes sense because they were brought over from Europe in the 1880s. The biggest takeaway I want everyone to kind of focus on though lies within that black circle. And this is just to illustrate that there's a strong overlap of brook and brown trout population and the potential for competition. Now we can zoom in a little bit closer into the driftless region. When you're looking at both these maps, just know the darker the color, the higher mean probability of occurrence of finding that specific fish species. So the map on the left just illustrates brook trout and the map on the right illustrates brown trout. And the black circle indicates just the location of where my thesis research is. So as you can see, the research site is located, there's a high mean probability of occurrence to find both species and the potential for competition in both these areas. So why is it really important to know this? It is important to know that thermoclasses can really influence the mean probability of occurrence of a fish species. And then this map here on the right illustrates a thermoclass map of the Wisconsin Driftless Region of summer mean temperatures in 2013. And this map shows 4 different colors, and those colors corresponding with what each thermoclass stream is indicated. And having the darker the color, the colder the stream. So dark blue meaning cold water stream and red indicating a warm water stream. And then the black circle is again locating just where my research is. And although you can't see where my streams, like it just looks white right there, mine are both considered cold water streams. It is really important to know where our cold water streams are and how to preserve those natural areas from changing thermal temperatures from climate change that will directly affect our trout populations. So, what is the purpose of conducting my specific research project? Well, I was lucky enough to be employed by the Wisconsin Department of Natural Resources in 2016, just as an LTE fisheries technician, and I've been working them for a few years. And then in 2018, a brown trout removal project was developed and planned to be started in the following summer field season. So since it's a multi-year project, there became a huge opportunity for me to take advantage of and conduct a graduate research project on the first 2 years. Next, in this research can assist in answering more questions for fisheries managers within the department and other agencies. And then this is where the knowledge gap was found within the driftless area. There's little known knowledge on interspecific competition between brook and brown trout, and there is a need to get more detailed understanding of food web interactions between brook and brown trout within the driftless region. And the reason why this project is being conducted is because the DNRS had conducted other successful brown trout removal projects. And one of those projects is on a stream near my research called Seas Branch. And to this day, it's sought after by anglers specifically for fishing brook trout. And in the brown trout removal section, to this day, there has been no reintroduction. And it's been several decades now, and no brown trout have come back into that area. So this has brought me to my 2 main objectives. My first one is to evaluate thermal distribution, habitat use, diets, and the interspecific competition between brook and brown trout. And then my second objective is to determine the carbon and nitrogen stable isotope signatures within the food web, and then evaluate the changes after we remove the brown trout out of that stream. To take and do this, I need to follow a very specific study design called the before and after control impact design, or BACI for short. And what a backy design requires is a reference stream. So I picked one where I did not do any brown trout removal, and an impact stream where there is brown trout removal. And then both these streams needed to be assessed before and after the brown trout removal process actually started, so you can take and do an analysis on that. And then it also accounts for the variability from year to year and sampling site to sampling site, which I'll get a little bit into more further in this presentation about how many sampling sites I ended up having. And the real big purpose of using this backy design is to evaluate whether or not brown trout has changed the stream food web, and then to estimate the magnitude of the other stream organisms being affected by brown trout. And then, okay, there we go. In reference to my first objective of determining interspecific competition of brook and brown trout, you first have to understand what interspecific competition means. So interspecific competition is just evaluating two individuals of different species competing for that same limited resources. So that could be either food, shelter, or reproduction. My research is able to evaluate that because we are removing brown trout out of my impact stream. And then along with my first objective in evaluating the thermal distribution, I can do that by using temperature loggers and then electrofishing surveys. And then with my final objective of determining carbon and nitrogen stable isotope signatures within the food web and then evaluating changes after brown trout are being removed, this can be done through stable isotope analysis. If you don't know what stable isotope analysis is, it is in the environment, we have carbon-12 and nitrogen-14. In my research, I'll be looking at stable isotopes that are slightly heavier, which are the carbon-13 and nitrogen-15. The carbon-12 makes up 99% of the carbon in our world, and that remaining 1% of carbon is in the heavier form of the carbon-13. And that carbon-13 has one extra neutron and can help track the flow of energy throughout the food web. And then similarly with nitrogen-14, makes up 99.7% of the nitrogen in our world, and that remaining 0.3% of nitrogen is in the heavier form of nitrogen-15, and that also has one extra neutron, and it makes it easier to track the trophic level of an organism. So with trophic level meaning the relationship between the consumer and its primary source of energy it's consuming. So using stable isotope analysis can give a greater understanding on food web interactions and a different movement patterns in fisheries ecology. And then kind of to take and give you a little bit more perspective of what stable isotope like graphs look like, I'll be completing a graph similar to Hershey in 2017 here shown on the left, where the delta carbon-13 is on the x-axis and the delta nitrogen-15 is on the y-axis. And I want everyone just to kind of focus where that black circle is. Again, this is the top consumer and should have the highest level of nitrogen-15 and having expected trophic levels of other organisms below within that food web. And if you don't know what piscivore fish are, it just means fish that usually feed on other fish. And then with my research project having the component of brown trout removal, this is where I'm looking for trophic shifts throughout my sampling site and sampling years, is all the organisms that are under the piscivore fish. And then going a little bit closer into where my actual project site is in reference to the backy design I previously explained, with the impact stream being Mapledale Creek, where brown trout are being removed, and the reference stream being Cook Creek, where there's no brown trout being removed. And the bright green color is just to indicate the Wisconsin state-owned easement or property, so you can see that a lot of it is actually owned and has state easement on it. And then both these streams are just located 3 miles outside Brook, Wisconsin, if you know where that is. Not too far from La Crosse, only about an hour commute. So on my impact stream, Mapledale Creek, there are several days spent removing brown trout from the entirety of the stream length. So we spent in 2019 and 2020, and we've begun now again in 2021 to start removing brown trout out of Mapledale Creek. The brown trout were removed and were collected by means of electrofishing and then relocated below the PL structure, earthen dam. And then this project site was chosen because of this huge structure there, the earthen dam barrier. This ensures that after the removal of brown trout are relocated downstream, they will not have the opportunity to get back into the impact stream of Mapledale Creek. And then that earthen dam does not have a screen on it, but we do believe that it is largely impassable just based on how high up it is, and it's really hard to take and jump into it. And then the flow itself is very, very strong. And since we ended up clipping about 600 adipose clips off of brown trout and then relocating them below this earthen dam, we have yet to see any brown trout with adipose clips back in Mapledale. So we still have yet to see any of them try to like get all the way up in there and then back into Mapledale Creek. In the summer of 2019, just under 15,000 brown trout had been removed from Mapledale Creek. As I said before, 600 had been clipped and then relocated below that earthen dam. And the remainder of them were frozen and then given to the Raptor Education Group in Antigo, Wisconsin. I think we're well over 200 pounds of brown trout we've given to the Raptor Education Group, just because of how high the populations are of brown trout in Bishop Branch that connects to Mapledale Creek, that it doesn't really make sense to take and keep populating that when it's already heavily populated with brown trout. And they go to a good cause. And then in the summer of 2020, another round of almost 3,400 adult brown trout were removed. There were a lot of YOYs were removed, but we just didn't count them. And then what is really worth knowing was in 2019, we found about 570 adult brook trout, and then in the entirety of Mapledale. And then in 2020, there was almost 4,000 adults found in the entirety of it. And now that we're starting it again here this last month, we are finding even more brook trout, and it is phenomenal to see how well they've rebounded. So getting a little bit more into my sampling sites. So within my project site, I actually have 3 sampling sites on my reference stream, Cook Creek, and then 4 on Mapledale Creek. So I have 7 in total. And then in each sampling equation, there is a population estimate that is being conducted, and that's just when you have a block net on upstream and then downstream, and then you electrofish going upstream multiple times to take and do either 2 or 3 passes to take and collect as many fish as you can to see what the population is within that segment, and then you can estimate that for the rest of the, Mapledale Creek or Cook Creek. And then I also conducted habitat surveys for each sampling site, which records like the types of vegetation, substrate, flow, water velocity, riparian zone within the first 50 yards of the stream bank. All that kind of habitat stuff was collected in each of these sampling sites. And then finally, samples were taken from each site for macroinvertebrates that are indicated here in dark blue, macrophytes, which are indicated in the light blue, Periphyton is indicated in white, the fine benthic organic material that is indicated in orange, and then of course finally my fish samples that were collected throughout each sampling site that I needed for stable isotope analysis. So a lot of things going on in just one sampling section. So some of my preliminary results that I've found so far, these images are just of conducting the first 2 years of field sampling for my research, that there are a few predictions that were thought to happen for my objectives. First is that after brown trout are being removed, brook trout would shift their habitat use, diets, and thermal distribution into the areas that brown trout once resided. So far in electrofishing in 2020, brook trout have moved into those areas where brown trout once resided. And then data is still being analyzed to confirm like how many are moving and where are they moving to. So that's still being done, but we can just in general electrofishing and removing brown trout, we can see how many brook trout are in areas that we never found them before. The graph here on the left is just to illustrate the 5 temperature loggers that I had within Cook Creek. And for the most part, they follow all the similar pattern. But it's just kind of interesting to see how when I show you the next slide, how this compares to Mapledale Creek. And it seems like Cook Creek is just running slightly cooler, which is a little bit surprising comparing their data of how many brook and brown trout were found in Cook Creek compared to Mapledale. There wasn't as many brook trout within Cook Creek. So it's just interesting that colder temperatures, you would think they would have more brook trout, and they didn't have as many. And then the graph to the right is just illustrating the catch per mile for the entirety of Cook Creek from years before and during this project. And as you can see, the overall brook trout population has been slowly decreasing, and brown trout have been continually increasing. And then this again is the stream where I didn't take any samples for, well, I only took samples for stable isotope analysis, but there's no brown trout being completely removed from this stream. And then going into Mapledale, to give you a little perspective what's going on in Mapledale, I ended up having 7 temp loggers put on Mapledale. As you can see, the temperatures in general are running a little bit hotter than they were in Cook Creek. But what is really interesting to take and see is if you see that orange line that's kind of like dead center around 50 degrees, that stays consistent through the entirety of the 2 years that I've had the temp loggers in Mapledale. And there's a spring that is coming right out of the hillside, and that is just constant same temperature. And it's just incredible to see how strong it holds through the winter and throughout the hot days in summer. And then the graph to the right illustrates the catch per mile for the entirety of Mapledale Creek from years before and during this project. And as you can see, the overall brook trout populations have slowly increased, and brown trout younger the year has increased, and then age 1 and older brown trout has decreased in the following year. The reason why we think that I think brown trout younger the year has increased from 2019 to 2020 is probably just because we're removing a lot of brown trout out of there, and you can't really get all the year classes in one shot. So I'm feeling as we continue, as the project progresses, you're going to see lower and lower numbers of the YOYs, the young of the year of brown trout. And then moving on to my invertebrate samples that I took, that I made very beautiful graphs for. And these images are just invert composition percentage graphs, which just shows you the percentage of the family within the sample that I took. So what's just really interesting to see is how some of these sites have higher representations of certain families in 2020 compared to the same site in 2019. And then some of the other families have a lower representation from year to year. So just some highlights of the years and the sites that I did. I ended up finding 28 families in 2019, and then about 1,300 individuals counted. And then in 2020, there was a total of about 27 families found, and almost all the same. There's a few that are different between the years. And then approximately 4,000 individuals were found. So much, much higher aquatic invertebrate composition in general in 2020. Some things just to kind of be aware of that maybe might have influenced my invertebrate numbers was in 2019, we had a little bit more flash flooding that happened over the summer, and one happened about 10 days before I had to take my samples. So it's possible that their numbers are a little bit lower because of that, but usually some aquatic invertebrate populations kind of rebound decently fast. So, but it might have influenced that. And then in 2020, we really didn't have any kind of flash flooding at all. So they just had time to grow and thrive and be happy. So that might also play a factor to why those numbers are much higher. Just really interesting graphs to see the differences in the different families and how they changed from year to year. So moving on to stable isotope analysis results. So with stable isotope analysis, what I want you to remember from the previous slide is that the delta carbon 13 is on the x-axis, which tracks the flow of energy throughout the food web. And the delta nitrogen 15 is in the y-axis that tracks the trophic level of the organism. And as you can see with the Cook Creek data analysis, brook and brown trout are showing the highest nitrogen 15 stable isotope signatures within the food web. And these were taken in 2019. I'm going to be comparing this to Cook Creek in 2020, and then also comparing this to all my Mapledale. So I'm going to end up having 4 graphs that look very similar to this, and then comparing all 4 of them together. And then what's interesting to see is brook and brown trout are showing that highest level of nitrogen 15 stable isotope signatures. And then what's also noting is that the leech family, I'm going to say this wrong, but I'm so bad with Latin. The Latin name for family for leeches Also, as you can see, it's a green circle that's above by the squares where all the fish samples. They are also up there, and what's just cool is just because they are feeding off the fish. So of course their stable isotope signature is going to be right in line with them. I just find it very interesting that it's hanging out with the fish. And then the rest of my samples taken for aquatic invertebrates, plants, and paraphyton all kind of follow the typical trends of the food web, that aquatic invertebrates are going to be lower than the fish, because the fish feed on the aquatic invertebrates, and then the aquatic invertebrates feed on the plants, and the plants and they also feed on the paraphyton. So it makes sense how it kind of goes in a downward slope motion. And then what I will be looking for is the differences between these types of samples from year to year, and then between Cook Creek and Mapledale. So what do I have kind of left to do? Well, I'm working on final data analysis, which is surprisingly a lot. And then I'm also going to be finishing up writing my thesis, and then preparing for my thesis defense. And then hopefully working on a couple different publications for my research. I'm hoping to be able to pull out a few different publications, but we'll see where that goes. First step is to finish writing my thesis and do my thesis defense. And then from that, I want to take and just kind of give a highlight summary of my presentation. And in summary, just understanding the value of Wisconsin's only native streams trout species being the brook trout, and the effects of climate change can have on cold water streams. Secondly, is how research can assist in gaining knowledge on cold water stream food webs, and then the underlying factors of brook and brown trout interspecific competition, the influences of groundwater tributaries can have, and the utilization of stable isotope analysis for measuring food web energy and trophic level within a food web. And then last but not least, understanding the importance of laying down more research for further brook trout restoration projects. And Caden's talking. And then that's my presentation, and I will take and leave you with that spring that I was talking about earlier throughout my presentation, just coming out of the hillside. It's just, the water is just dumping into Mapledale. And I'll take any questions that anybody might have.
Hey everyone, I'll be taking over the meeting from here to now.
Yeah,
Hey, Mr. J.
Sir, Mr. J. William, please quiet down.
What page?
Hey man, please sorry about that. Hey,
hold on a sec.
Wait, I have a question.
Shoot.
I have a question.
yeah, Mr. J. William.
No, all right. So what about this whole nature life?
What do you mean? What about it?
Sorry, I didn't hear it. Like,
No, I was
halfway
asking Krista you a question.
Yes.
All right, so
I wish.
Hey, sorry about that, guys. I think we had some folks who were hopping on trying to wreck the meeting.
I was gonna say, I have no idea what he was trying
um,
to ask either. He like cut out halfway through.
Yep. Yeah, I'm not sure what's going on there, but I don't know. Jason, you want to kind of monitor some of that, and if you see anything that looks a little fishy,
I relocked the room.
maybe ask the folks to leave. So, there were a couple of questions in the chat box. I think
Brian, how old are you, my man? What are you doing? You want to mute real quick, Brian?
How many tiger?
Here, hold on one second.
How many tiger trout did you find?
Oh, so I wrote a note in.
Yeah, I found a couple back in my day.
A lot.
Back in Really? How many?
Probably close to 30 in Mapledale. We haven't found any in Cook Creek, but that's the one perk of doing the brown trout removal project is, yes, I have these 7 sampling sites, and I might find a few in the sampling sites, but we're doing the entirety of Mapledale, so I get to see a lot more tiger trout. So I think we've been probably close to 30, maybe more. Granted, we're not marking them either, so they could be the same tiger trout after year and not knowing it.
That's interesting because Cook Creek had a pretty good population of tiger trout about 15 years ago.
Yeah, I haven't found any yet, but there's another graduate student out of UWL. She's doing occupancy modeling between brook and brown trout and looking for tigers, and Cook Creek is on her list. So it'll be exciting to see her results to see if she finds the likelihood of finding more tiger trout in Cook Creek.
It is yeah, that'd be interesting to follow for sure. Paul had a question. He was wondering how much is global warming going to affect the thermal classification of the streams in Wisconsin? I know when you show those maps up there with the, the blue and the green, and then things turn into red. Is that are folks monitoring that right now? And are we seeing trends yet, or is it?
Yes.
Is that data not available
So
yet?
like, I'm definitely not an expert at it. There's definitely some higher up people in the department that are way more expertise. This is what they do for a living on monitoring, but there are a lot of people within the department, and I know with USGS, that are monitoring a lot of these trout stream temperatures and seeing how things are starting to warm and change. At least in my last 5 years seeing these trout streams, I can tell you that I've used to see brook trout in my first year, and they're not there anymore, where there's good healthy populations, and you can see the temperature is warmer. So whether that is a distinct factor of the issue, that's one of the main points in my research, is that temperature might not be dependent on it, that it might be more about interspecific competition, that brown trout are generally more aggressive, they grow faster, they grow bigger, they can spawn out more eggs, they can tolerate a lot more harsher conditions, they feed on that same type of food source as brook trout, so they might just be the bullies of the trout stream and just pushing these brook trout out of their native range because they don't have anywhere else to go.
Yeah, it's certainly interesting. I know it always makes me think of the research that John Lyons has done and some of the presentations that he's given in recent years, where he shows
Thank you,
some of those
Kristina.
graphs where we're expected to lose 2/3 of all of our brook trout water over the next 50 years, and like 1/3 of our brown trout water. And it's going to be really important for us as Trout Unlimited, if we're going to work towards finding ways to mitigate that at some level. We're going to need to understand, like you say, how the brown trout and the brook trout are interacting in these streams, and maybe we need to look at changing some of our the way we do restoration work, depending on what species that we're trying to improve. So, which I guess leads into the next. There was a question here: so what are some potential applications of this research to the management of brook versus brown trout in Wisconsin? Kristina, do you have any insights into how this might be used to guide management decisions?
That might be more of a question for my boss, but just in my personal opinion, not necessarily like in just the research that I've seen, I have a feeling that if this gets published and works out well, and they see a strong rebound of brook trout population, especially how they did in Seas Branch, I could only imagine how many more brook trout restoration projects where there's those giant PL structures or another really good barrier dam, and they have a good brook trout population above that area, to keep promoting more restoration projects like this to preserve these brook trout in their native range and keep them from being hindered from brown trout.
Yeah, absolutely. So here was kind of a fun question, and I think you piqued somebody's interest when you mentioned that some of the brown trout got donated to the Raptor group. Somebody was curious if there was a specific size that they were looking for, or did they just take all the fish and kind of ground them up? Or
I don't know necessarily what they did with them. I'm assuming that they probably ground some of it up, but maybe the bigger ones that were on top, that they just thawed out and probably gave it to them. I know I've been to a couple eager centers, and you just see them, hucking whole fish at them, and they love it. But they were solid. I brought several coolers to Anago, and I was like, they're gonna be happy as a clam.
That's great. Yeah, that's a whole new meaning of Friday night fish fry, right?
Right.
Yeah. Somebody asked, can you explain what exactly electrofishing is?
Yes. So electrofishing is where there's several different methods. So like the two methods that I use primarily backpack shockers, just because this is a smaller stream. So you're able to take and use a backpack shocker, but there are stream shockers. There are mini booms that are on our bigger rivers, like the La Crosse River or the black parts of the Black and Coon Creek, like some of these larger waters that are completely not wadable. And then there's also the maxi boom, which you see on the Mississippi River. So there's several different forms of electrofishing, but like the primary one that I used was backpack shockers. And literally it's a backpack that you would wear, and it has a small battery on the back, like you would have for like a motorcycle, just a 12-volt battery. And that battery is connected to the backpack shocker, and that has a whole bunch of electronics that are above my pay grade of understanding the ins and outs of how all that electronics work. But the electricity is converted in that box to DC pulse. So it's direct current pulsing from the backpack, and it goes into a probe. And then then the probe goes into the water. And then there's also a rat tail, we call it, but it's another piece of equipment that comes off of the backpack shocker that drags behind us that has metal on it. So to basically complete the circuit between the probe and the rat tail, because they're both metal. And then basically what happens is when you have the probe in the water and you have the backpack shocker on, the electricity goes through the water, and depending on how high it is, you can basically stun the fish, and they turn. They're swimming there because they're scared of you. They don't know what that person is doing in the water, and you turn on the electricity, and they basically turn towards your probe, and then you can take and catch them, and they rebound really quickly as long as you don't have the electricity too high. I hope that answers your question. So
yeah, I think so.
Electrofishing.
Yeah, I've seen it done a couple of times, and as an angler, it's pretty humbling to watch somebody go through some of these streams, and it's amazing to think of just to see how many fish are actually in these holes in a stream. So that's pretty neat to see. Here's somebody had a question. I think one of the graphs showing the number of fish showed a pretty big spike in the number of sculpin in one of the creeks.
Yes.
Was that correct?
So
Do you have a theory behind that?
I can go back to that graph so everyone can see it. Let's go if I can. Oh gosh, hopefully this doesn't take too long. Okay. So yeah, so with the slimy sculpin population, and this is because I'm still working through my data analysis. My first gut response is that slimy sculpin population is increased because there's not as many brown trout in there to eat them. So that's my general thought of why the slimy population, but they also just could be having a really good year. That Mapledale area has had such a strong slimy sculpin population for a number of years. They've taken that population and stocked them out to other parts of the state. But that's just my first gut response to thinking of why that population is so high. It's just because they're just rebounding really well because they're not being preyed on by brown trout anymore.
Yeah, that makes sense. Here's a question. Is the brook trout a preferred tourism species, or is it just a matter of prioritizing the native species?
So, and that's, it's depending on who you talk to as an angler. You know, some anglers are really into brown trout and they don't really seek out brook trout. Brown trout generally get bigger, so some people are really into just getting bigger fish. Some people love going for brook trout because of how vibrant their colors are, and not even during breeding season. They just are beautiful fish to see in person. So it's just like, it just depends on who you talk to. I'm a fan of brook trout.
Yes, we at Trout Unlimited, we're obviously fans of brook trout too. You know, it's the native species, and we're a little bit partial towards them. Obviously, we recognize the value of having brown trout around, and you know, the resource that is as well, and the ability to draw anglers into certain creeks that maybe they're a little too warm right now to harbor, populations of brook trout like they did 200 years ago. So the brown trout maybe makes a suitable replacement in some of those situations. But yeah, I think from our organization's perspective, we look to protect the natives first, and then a wild species like a brown trout second. So if we were going to rank them. But here's a question: do you expect a larger brook trout to be more abundant now that the browns are gone from the water, or do you think the size structure of the brook trout are probably going to stay about where they were?
I'm still working on size structure graphs in my data analysis, but we've already seen that they've been bigger. They're just growing and being fat and happy and living longer. So without the brown trout in there, they're able to at least not be picked away right away and be able to take in, kind of compete legitly.
Interesting. Yeah, that's kind of interesting, because I've heard people talk about, like, you think like the biggest brook trout in the world are like up in Canada, and I guess I've always heard that the reason they grow so big up there is because there's northern pike. There's like a big predator. So like if you don't get really big, like you're not going to survive. But here we almost, like, your research is almost showing the opposite there, where like you're taking away their maybe one of their main predators. Well, I guess maybe not their main predator in the stream. I mean, there's a lot of other predators around here in Wisconsin, but you take away one of the predators anyway. But they're just being, like you said, they're just happy.
And that's the thing that you have to think about too, is so in Mapledale and Cook Creek and even Bishop Branch below it, like in general, the driftless area, all our streams are chock full of trout. Like you put your pole in any cold water stream, you're probably going to catch a trout. So it's just these populations are so high and starting to remove some of that pressure of having basically a lot of fish, and not like you have a lot of food there, but it's not enough to support the amount of fish you have in that stream. Might also be helping relieve the pressure and be able to get fat and big and happy.
Nice. I must be doing something wrong, because I've gone fishing a number of times out there in the driftless, and I've not been able to catch a trout. It can be humbling, but there are definitely good days too. So, and you're right, the streams are full of fish, so which is a good thing. So, let's see here. How much influence have you found water temps have on the brook trout?
And that's one reason why I have those temperature loggers out to kind of compare the temperature logger data to my PEs, the population estimates that I took in each sampling site to see, okay, so in this sampling site, we found so many brown trout, so many brook trout that has this type of temperature generally ranging throughout the year. And now we removed the brown trout. Is there more brook trout there, or is there less brook trout there? Have they moved into that area? Can they thrive in that area? And it seems like the temperature doesn't really mean anything. They're moving into areas that we've seen like brown trout once were residing, and they're happy in the headwaters. They're happy in the little bit of warmer water in the more downstream end. They don't seem to care. I think they're just happy that they're not being picked on anymore.
Yeah. Are there any additional streams slated for brown trout removal that you're aware of?
Not at least in the counties that I am kind of associated with working for the department. I believe it's possible in the northern section of Wisconsin that have a lot of trout water up there. They might be doing some, but I'm not aware of any going on right now. But I hope that there is going to be in the near future.
Okay, that's good. I think somebody asked a question about says, since most of Wisconsin brook trout are native, how is it not native in the southern part of the state? I'm not sure that's a correct statement, because they're native everywhere in Wisconsin, correct?
Correct.
Yeah.
it was me. I was looking at the map you showed, and there's a little brown stretch of the map at the southern part. Okay. That's dark brown.
Yes. So on that map, technically there is like a small section that they're not native to, but I don't think it's enough to take in really. To me, when I think of brook trout, they're native pretty much primarily through the entirety of Wisconsin. It's just like a very small section. I'm guessing maybe because it's more agricultural and they never were found in those areas. Maybe it was warmer down there in general, and they just never got moved into that area right away. I'm not exactly sure.
All right, thank you.
Yep.
Yeah, it kind of looks like, I'm not exactly sure what watershed that would be, but you're probably right. That's probably just a warm watershed down there in that little stretch, and there just aren't any, never were any, like historically, any brook trout found there. So
yeah,
it's interesting to see the anomalies show up on the map like that, though. Let's see. I think I want to say we've covered all the questions. Does anybody else have? Oh, somebody asked if you take any stomach samples. Do you ever like pump the stomachs?
Yes, so that's part of, it wasn't generally a part of my original thesis that I was going to end up doing, but with my 2019, we ended up removing the stomachs, and that has already been analyzed, and I just have to take in, done the lab work to see how, which invertebrates were in the stomach samples. I just have to take and figure out what's actually going on. My boss, Kirk, ended up doing it on his free time. He wanted to see what they were eating, so. But he gave me the data, so that's extra stuff for my project.
Perfect. And what were they eating?
I haven't even opened it yet.
All right.
I'm guessing a lot of scuds. That's what I'm guessing, because I had a lot of gammarus in my samples. And
a lot of scuds.
Chironomids. I'm guessing. That's what I'm guessing, but
yeah,
we'll see when I open it up.
Yeah, well, in that part of the driftless, there's probably a very good chance that that's what they were eating, I would say. So I think we're getting close to probably the end. I just want to, on behalf of the state council, I want to thank you for coming on and sharing some of your preliminary data with us. We're super excited that folks like you are out doing this research, and we want to make sure that it continues and that we keep building on the knowledge base, and that we can, continue to do all we can to make sure our native brook trout are thriving here in Wisconsin. So.
No, I really appreciate you guys helping with part of the funding. Stable isotope analysis is not cheap at all, and that helped me with a lot of samples. So I am very grateful for you guys helping me out with that.
Somebody else that I just saw a question come in was asking if you think the brook trout favor one particular kind of habitat. If you see them gravitating more towards a particular habitat as opposed to necessarily maybe a water temperature or a food source.
And I did take habitat surveys to take and kind of evaluate to see if they were, but we found them throughout Mapledale and Cook Creek. So I don't think any type of habitat really was a huge factor, but I'm only doing it on 2 streams to say that that's a hard fact. I can't say that for sure.
Yeah, that makes sense. Excellent. Well, I think the invitation is open. If you would like to come back when you have finished crunching the numbers and you have, some better data to present to us, or some analysis to present to us, we'd be happy to have you back on at a future date. So yes. Thanks for spending some time with us tonight. We really appreciate it.
Thank you so much. I really appreciate it.
Awesome. Thanks everybody. Have a good night. Get out there and fish. It's prime time for trout fishing in Wisconsin right now. So enjoy it. Thanks.